Good morning, students. Today we are going to discuss about a new chapter number 8, Cell the Unit of Life. This chapter is very important for board theory examinations and as well as for competitive examinations like NEET and KCET. Right? So let's enter into the chapter. Once have a look into your surroundings. You may absorb both living and non-living things a lot, right? You may have wondered and asked yourself even some questions like this. What is it that makes an organism living? Or what is it that an inanimate thing does not have which a living thing has? The answer to this is the presence of the basic unit of cell. So the basic unit is nothing but here, cell. Cell in all living organisms present and makes the differentiation between living organisms and non-living organisms. All living organisms are composed of cells. Some are composed of single cell like paramecium, euglena, amoeba, etc. These kind of organisms which possess a single cell in them are called as single cell organisms, right? While others like humans and other animals like elephants, horses, cows, buffaloes and plants, etc. composed of many cells, more than one cell in them. Such kind of organisms are called as multicellular organisms. Got it? Now see on the screen you would able to see a beautiful human cell, right? First of all, my question to you is, what is a cell? How can we define it? Now let's enter into that concept. Unicellular organisms are capable of, first point, independent existence and performing the essential functions of life. Anything less than a complete structure of a cell does not ensure independent living. Hence, cell is the fundamental structural and functional unit of all living organisms. Understood? So, when should we consider it as a cell? It should possess two basic things. First one is independent existence, then performing basic essential functions of life. If anything is less than a complete structure of a cell, it shouldn't be called as a cell. Make sure this point must be clear to everybody. Understood? Now, I am going to introduce a great scientist here. You can see on the screen. Under early contributions, Robert Hooke, the first person to see cells. He was looking at cork and noted that he's a great many boxes in the year of 1665, and he popularized as a scientist who discovered non-living cell in a cork piece and named to those as cells. But later I would like to introduce one more scientist. The scientist's contribution you can see on the screen. Anthony van Leeuwenhoek observed living cells in pond water, which is called animalcules, in the year of 1673. It means later to Robert Hooke. The contribution goes to credit goes to Anthony van Leeuwenhoek in the early contributions. So now you see the screen about Robert Hooke. One more important point. He studied a cork, a kind of tree bark, named these structures he saw as cells because they remind him of the small rooms monks slept in. Might be you have seen a small rooms where monks slept in. Such kind of rooms were absorbed by him in the cork piece, which is a small tree bark. He named it to those as cells in the year of 1665. Comes under early contribution. So make sure this point must be clear to everybody. Right now, another important point, another important scientist's contribution, Antony von Leeuwenhoek, early 16th century. So as we discussed just now, he discovered the cells in the year of 1673. You can see in this present slide 1673, later to Robert Hooke, right? So now students, let us see the important information regarding Antony von Leeuwenhoek. So here he is a Dutch tradesman and scientist, studied pond water and sold milk and cement, named to those organisms as animal cures. Hence, he popularized as father of microbiology. Father of microbiology. You know microbiology. A study about microorganisms called as microbiology. It is a branch of biology, right? Next, I would like to introduce one more scientist. Antony von Leeuwenhoek only, and the special contribution of Antony von Leeuwenhoek is here has to be discussed, and he is the scientist who used a microscope first time. That's why he is also popularized as father of microscopic and father of microbiologists and father of microscope. And here he is the scientist in the year of 1670, first person to observe cells under microscope which are alive. He was looking at the microscopic organisms found in a pond water. You can see the organisms on the screen. Different kinds of organisms are present on the screen. All these organisms are noticed by Antony von Leeuwenhoek in 1676 exactly. They are all live and noticed all these organisms in a pond water, right? So used microscope first time. And now coming to another scientist. One more point I would like to bring to your notice regarding Antony von Leeuwenhoek. And Leeuwenhoek used a simple handled microscope to view pond water and scrapings from his teeth. So these are all important discoveries can be noticed regarding Antony von Leeuwenhoek. You can see the microscope on the screen right now. Next, coming to another scientist, Rudolf Virchow. He's a German pathologist. He is popularized as father of pathology. Discovered that all living cells come only from other living cells. It means nothing but he discovered cell division. So cells are born from parent cells or pre-existing cells. This phenomenon scientifically proved and called as cell division, which are already got existed. From those existed cells, new daughter cells are borning through a scientific method called as cell division. So this was noticed by Rudolf Virchow. This person is popularized as father of pathology, right? So you can see on this present screen how the cells are dividing. In the year of 1855, the German scientist Rudolf Virchow stated that every cell originates from another existing cell. Virchow demonstrated the first evidence of cell division. So he is the first scientist, one who demonstrated cell division firstly. So what a great contribution done by him, right? So now let us talk about one more important point about him. You can see on the screen how the cells are dividing, how the cells are forming from the pre-existing cells. This is also a contribution, nothing but about Rudolf Virchow. Next, coming to another important point, Robert Brown. You can see Robert Brown's worksheets on the screen right now. Whatever he described, whatever he discovered, he have noted very clearly in his worksheets called as Robert Brown's worksheets. They are also available right now in the world. Next, let us see the contribution of Robert Brown. He discovered the nucleus in the year of 1833. And what a great contribution by Robert Brown to the science field. And he observed one important point. And while observing a collected plant cells, he discovered an opaque spot in the middle of the cell and named to it as nucleus. See, findings were published in the Linnean Society paper in 1833. He discovered that its structure was directly related to the cellular tissue. So, great contribution done by Robert Brown. Discovered the nucleus. You can see on the screen nucleus structure, which is discovered by Robert Brown. Next, see one more point regarding him. He is an English scientist. In the year of 1830, built an improved microscope. After that, whom? Anthony van Leeuwenhoek. Next, used it to observe cells he had stained with various reagents. First, to observe the nucleus of cells. Who is the person? Great scientist Robert Brown. You can see on the screen scientist and his contribution to the science field. Next, another scientist I am going to explain you now, cell theory. Okay, what is meant by cell theory? Here we have to discuss about two great scientists and their contribution. First and foremost, I would like to introduce a scientist in the year of 1839, a German zoologist named Theodor Schwann conjectured that all animals were made up of cells. Right? Schwann also co-founded the cell theory. You can see the scientist on the screen right now. Now, let us see one more important point here. Here, earlier to Schwann, one more great scientist in the year of 1838, a German botanist named Matthias Sheridan conjectured that all plants were made up of cells only. Whatever the plants you are seeing in the nature, either irrespective of size, height, everything like herbs, shrubs, trees, whatever it is, all plants are made up of with cells. Sheldon is a co-founder of cell theory. So you can see on the screen the scientist and his contribution. Next, here in the single frame you can see three scientists together. In order, first scientist. In the year of 1838, Matthias Sheridan suggested that all plants are made up of cells. Just now we had a discussion. Next, after that, in the year of 1839, Theodor Schwann suggested that all animals are also made up of cells. Whatever the kind of animal you observe or we observe, all those animals are also made up of with cells, irrespective of their habit and size and even feeding habits. All those animals are made up of with cells, more than one cell. Next, later to Theodor Schwann in the year of 1855, Rudolf Virchow. What is his contribution? All cells arise from pre-existing cells by cell division. So here I would like to mention one important point here. First two scientists, a botanist and zoologist, just they discovered that plants and animals are made up of with cells, right? But they unable to prove scientifically how new cells are born from other resources. So now let us have a talk about one important point. This geologist Theodore Schwann, and in the year of 1839, a British geologist studied different types of animal cells and reported that cells had a thin outer layer, which is today known as plasma membrane. He also concluded that based on his studies on plant tissues that the presence of cell wall is a unique characteristic of a plant cells. Right, see what a great contribution. Being a geologist, he also contributed, he also studied about plant cells, and he concluded that plant cells have a unique characteristics, that is nothing but presence of cell wall on the surface. On the basis of this, Schwann proposed a hypothesis that the bodies of animals and plants are composed of cells and products of cells. Right. Next, Sheldon and Schwann together formulated a theory called as cell theory. However, cell theory did not explain as to how new cells were formed. Just now I just told you, right? Next, continuation to the third image belongs to Rudolf Virchow. In the year of 1855, firstly explained that cells divided and new cells are formed from pre-existing cells. It is scientifically called omnicellular e-cellular. It means cells are formed from pre-existing cells. He modified the hypothesis of Sheridan and Schwann to give the cell theory a final shape. So cell theory as understood today as, let's see what points are included under cell theory. I would like to say the first point here. All living organisms are composed of cells and products of cells, and all cells arise from pre-existing cells. Right? See on the screen about both scientists, Mathis Sheldon and Theodore Schwann. And they concluded only one point. What's that point? It is nothing but all living organisms are made up of, are composed of cells and products of cells. Right? But cells arise from pre-existing cells. It means scientifically called as omnicellular e-cellular. Is proposed by Rudolf Virchow. See the diagrams or images on the screen. Three scientists together present in one single frame. So three scientists formulated a theory called as cell theory. Understood? So according to cell theory, I would like to tell you the highlights. First point, see on the screen. All living organisms are composed of cells and products of cells, which is a common hypothesis for both Mathis Sheridan and Theodore Schwann, 1838-1839 respectively. Next, the second point, all cells arise from pre-existing cells according to the concept of Rudolf Virchow in the year of 1858. Now, students, I would like to say one more important continuation concept. It is nothing but an overview of cell. Okay, now earlier observed cells in the previous classes. In the previous classes you have observed the cells in onion peel or human cheek cells under microscope, right? And try to recollect those structures, how they are present. You please tell me. The onion cells, which is a typical plant cell, has a distinct cell wall, its outer boundary, and just within it is the cell membrane, right? The cells of the human cheek have an outer membrane as the delimiting structure of the cell. So what is the limited structure to the animal cells? It is nothing but plasma membrane. In the case of animal cells, plasma membrane. In the case of plant cells, it is nothing but cell wall, right? So below the cell wall, there is a thin membrane called as plasma membrane. Next, inside each cell, there is a dense membrane bound structure called nucleus. Who discovered the nucleus? In this class we had a discussion, nothing but Robert Brown, right? This nucleus contains the chromosomes, which in turn contain the genetic material DNA, deoxyribonucleic acid. Cells that have membrane bound nuclei are called eukaryotic cells, whereas cells that lack of membrane bound nucleus are called as prokaryotic cells. So this is very important point for theory examinations. Difference between prokaryotic cells and eukaryotic cells. Pro here means nothing but false. So false membrane or completely membrane is absent. So they are not having true membrane bound cell organelles, or nothing but called as prokaryotic cells. Eukaryotic cells, they are having true or real membrane bound cell organelles called as eukaryotic cells. So a semi-fluid matrix called cytoplasm occupies the volume of the cell. You just imagine like one classroom which is having 4 walls, that should be considered as a cell covered with a cell wall in plant cells, cell membrane in animal cells, but inside the whole class is occupied with a semi-fluid matrix. It is called as cytoplasm, which occupies the volume of the cell, right? The cytoplasm is the main arena of cellular activities in both the plant cells and animal cells. What is meant by arena? It is nothing but a main site. It's a main place for all cellular activities in both plant cells and as well in animal cells too. Various chemical reactions occur in it to keep the cell in a living state. That's why the cell always in a living state. Various different kinds of chemical activities will take place from cytoplasm itself. That's why it is a site for cellular activities, which makes the cell alive in state, right? Besides the nucleus, eukaryotic cells have other membrane bound distinct structures called organelles. In eukaryotic cells, distinct, it means clearly visible membrane bound cell organelles. Those membrane bound cell organelles are as follows, like endoplasmic reticulum, shortly called as ER, Golgi complex, lysosomes, mitochondria, microbodies and vacuoles. Whereas coming to prokaryotic cells, in prokaryotic cells we won't see clear cut membrane bound cell organelles, and these are such as ribosomes, a non-membrane bound cell organelles found in the cells, both eukaryotic as well as prokaryotic cells. Understood? So within the cell, ribosomes are found not only in the cytoplasm but also within the 2 organelles also they are present. It means ribosomes are present in the cytoplasm first place, and as usual they are also present within the cell organelles. The membrane bound structures which presents in the cytoplasm are called as cell organelles, right? In those cell organelles also ribosomes are present, not only in the cytoplasm. What are those? Such as chloroplasts in plant cells, mitochondria And as usual, we can also notice on rough endoplasmic reticulum. So there are three cell organelles. In those three cell organelles also ribosomes are present apart from cytoplasm. Make sure this point must be clear to everybody. This is important concept for competitive examinations like NEET and KCET. Next, animal cells contain another one membrane bound cell organelle which is absent in plant cells. Those are nothing but called as centrioles. These centrioles helps during plant cell division, during animal cell division, right? As usual, during plant cell division, there is a one more cell organelle which presents exclusively in plant cells or called as microbodies, helps in plant cell division. Make sure these 2 points are clear to everybody. Important for competitive examinations, especially for NEET examination. We'll repeat these 2 points once again. Name the cell organelles which presents in animal cells and plant cells respectively, helps in cell division during their cell division time. Okay, we'll tell you one by one. First and foremost, centrioles which presents in the animal cells, which helps animal cell division. This is also non-membrane bound cell organelle, presents exclusively in animal cells, helps in cell division, right? Here you can see some plant cells on the screen, and as usual you can see some animal cells also on the screen. In these animal cells only these centrioles are present. Then coming to plant cells which you are seeing on the screen, in these plant cells also there is a exclusively organelle called as microbodies. These microbodies are present in plant cells which helps in plant cell division, right? So these two are important points. Next, see the present diagram. This is very important concept. Cells differ greatly in size, shape and activities. For example, mycoplasmas. About mycoplasmas we had a discussion in previous classes in lower chapters like biological classification. So here students, a smallest cell, it is considered to be as a smallest living cell by scientists. Cell wall is absent and can survive without oxygen, right? So this is a smallest cell. Even the size is 0.3 μm in length. See, such a small it is. While bacteria could be 3 to 5 μm. So you can see on the screen, bacteria could be 3 to 5 μm. Next, the largest isolated single cell is the egg of ostrich. So ostrich egg is considered as the largest isolated single cell on the land. Next, among multicellular organisms like humans, red blood cells are absent, or about they are in red blood cells are absent in some organisms. Okay, very good. Like some insects, they are not having RBC in their blood, but RBC is present in humans blood and animals blood, and they are playing a major role. Red in color due to presence of a color causing pigment hemoglobin, and binding oxygen and carrying from one place to another place, right? And mixes with blood called as oxygenated blood. So these RBC are about 7.0 μm in diameter. A human RBC is about 7.0 μm in diameter. Next, coming to nerve cells, it is nothing but fourth diagram on the screen. These nerve cells, some of our longest nerve cells. Can I tell you one important point here? The longest nerve is present in our body. It starts from the hip region. It means from it means below the abdomen, hip region. From that through femur, knee, ankle, it spreads like that. So the longest nerve is called a sciatic nerve. This sciatic nerve is longest cell considered so far. Next, cells also vary greatly in their shapes also, right? So now you see on the screen they may be disc shaped, like the first one RBC, polygonal, that's the second one, columnar, cuboidal, thread-like structures, or even irregular in structure. Understood? Elongated, a plant cell can be seen here on the diagram, nothing but a trachid, which is red in color from last to second one. Next, mesophyll cells, round and oval shape. They are also plant cells. What is their shape? Round and oval shape. Next, branched and long cell, example nerve cell. In the diagram, fourth diagram, from starting onwards it is fourth diagram, branched and long cell. Next, in the diagrams list third one is nothing but columnar epithelial cells, long and narrow, long and narrow. Next, white blood cells, they are they're having a specific structure called amoeboid structure, because they do not have a particular structure like amoeba, since they are called as amoeboid structured cells. Next, round and biconcave shaped structures are nothing but RBC, first diagram, red blood cells. So these are all the different shapes of the cells. Hope you understood today's introduction and overview of cells and different scientists' contribution regarding cell and its discoveries. In next class, we will discuss regarding each cell organelle in detail. Thank you for your listening.